Electrical power.
Thermocouple temperature is at 17. Super is at about 10. Can we report the flow rate and volume for the last filter?
Yeah, so for filter S1, we had 0.14 liters per minute, which is lower than we got on the last microdive. The total volume was 2.77 liters.
Okay, so 0.14 liters per minute and
Yeah.
2.77 liters.
Yeah.
So our ROVs, Hercules and Argus, these are remotely operated vehicles. They are not submersibles with humans inside them. They're controlled by humans who sit in a control van atop the Nautilus. And all of our commands are transmitted down that long cable I told you about.
You're not on SPL.
Are you sure?
Or force talking.
Right. Hey Nautilus, or hey Nautilus, hey Alex.
To try what?
Can you do a ship move?
You want to ask Julie?
Let's go 5 meters at like 200.
5 meters
Yeah.
at 200, copy.
I think where currents and stuff are changing around on here. We're laying back in different ways.
Bridge nav.
Try and slide us
220.
over that way, but
Roger.
Can I get 5 meters at bearing 220?
I think it was
let's
200.
go 200. Yeah. Well, it'll be whatever.
As long as we
Either
get
one.
plenty of bag fluid, we also filter that
Close
onto
enough.
a Stervix, so we can use that as well. Yeah. Mm-hmm.
So one of the critical components of deep ocean robotic exploration is the ability of the Nautilus to hold position to really within a meter or less on the seafloor, on the surface. We do that via using GPS satellite receivers to give us our position, and then we use what's called a dynamic positioning system to basically direct a jet of water from the underside of the ship in whatever direction we need to apply it to counter the effects of wind and current and wave action on the surface. And what that means is we offer as stable a platform as possible on the surface from which to deploy these long cables to the seafloor. If we moved around too much, we'd actually drag the ROVs with us, and that would be a bad thing.
Oh, that's okay. 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 Flow rate still at 0.7.
Thermocouple temperature is at 13.8. Super is at 7.6.
They're passing chocolate around in here.
Yeah. Thank you to Jessica for the intense orange dark chocolate. It's a scientifically proven fact that the consumption of chocolate alters your perception of time.
Yeah, we just started to watch, I think, right?
Mm.
But I know.
Oh.
let's do another 5 meters on that same bearing.
Copy that.
Good.
Bridge nav. Yeah, one more step, please.
Has anything picked up out there, Josh?
No, I just think that as we're sitting here over time, you know, the currents change and the conditions change, and so our layback has changed because we're so deep that we're impacted by those
Okay.
small changes. And so even though the ship hasn't really moved, Argus is laying back a little differently just from the forces on the cable
Right.
and the vehicle. Yeah. So we're just adjusting to get in a little better spot.
Oh, I see.
It's
So
kind of a cool shot of Herc, but I want to get a little more behind Herc.
yeah.
Yeah.
So the ship kind of keeps its position, but as you said, the additional current, we can't correct the cable. That just experiences the force in the water column.
That's right.
Yeah. So one question we had, I was just going to throw out quickly while we're on that subject, is typically what's kind of the maximum wind speed that the dynamic positioning system can handle?
It depends a little bit on not just the speed, but the current and the wave conditions.
Uh-huh.
But generally speaking, kind of 25 is somewhat of a threshold. It can do 30 on some occasions, but
Right.
it kind of depends on those other factors.
Thank you.
Yeah.
So just for those of you on Nautilus Live joining us, we're getting a couple of questions about hydrothermal vents and what kind of environments they represent. So you are actually looking at a hydrothermal vent right now on Hercules' main camera. This is what we call a diffuse flow vent. This is where we don't see perhaps one of those tall chimneys you might be used to seeing on documentaries or on the internet. And this is a site where you've got the water is not coming out in a focused flow, but it's coming out over a more diffuse area. Typically that means that the hot vent fluid has mixed with more seawater. Now the seawater is at approximately 2 degrees Celsius. And so what that does is that kind of lowers the temperature of the mixed water that results. And what we're seeing, at least at the Seacliff hydrothermal vent site, is that it's these sites of diffuse flow of slightly lower temperature mixed water where we're seeing most of the biology. What you're seeing in front of you, that mass of kind of tangled white-coloured worms, really, those are the tubes of tube worms, Regia pisceci. And you'll see if you peer in as well, you'll see some other creatures in there. Lots of the tiny little almost spherical nodules you're seeing on a lot of the material. Those are little limpets. And then you can just see where the Hercules manipulator arm is holding that wand, and that wand goes down in the Hercules field of view, and see those almost like waving fronds in the vent flow. Those are another kind of worm. Also, it's called Alvinella pompeiana, which is a fancy Latin way of saying the Pompeii worms discovered by the submersible Alvin. If you
You
look
need to come a little wide on Argus.
Let's come a little wider, and then, yeah, Alex, let's do one more move. Let's do 5 meters, like 260.
5 meters at 260, copy.
Yeah, thanks.
Bridge nav. Yeah, 5 meters at bearing 260.
So another great question on Nautilus Live is, what's the typical pH value or acidity or alkalinity of these hydrothermal vents and the chemical composition of the vent fluids? I don't remember the pH value. Do any of you guys in the back row remember that approximately?
4 or 5, I think.
Okay, so
Wow.
5-ish.
mildly, well, reasonably acidic. In terms of the chemical composition of the vent fluids, if you were watching earlier, you saw us take what we call an IGT sample, which is an isobaric gas-tight sample of the vent fluids. This is a neat sampling method that maintains the sampled fluid at the same pressure it was when it was sampled at the vent, all the way back to the laboratory on board the Nautilus. And so we can actually sample that water without the gases coming out of solution. And we see a lot of hydrogen sulfide, a lot of methane are the principal constituents we're actually able to pick up with the equipment we have in the lab.
So data, the flow rate is kind of slow. It's been about 0.7, but it's slowing down to 0.4 more often. It's kind of struggling a little bit.
Got it.
And the temperature on the thermocouple is 19.6, and super is about 10.
For those of you wondering if these hydrothermal vent environments are the only environments in which we see tube worms, the answer is no. You can see them all over the ocean. I live in British Columbia, and so when I go snorkeling there, I encounter them in the tidal shallows. But what you're seeing here are some really specifically adapted hydrothermal vent tube worms that are able to withstand much greater temperatures than normal, or let's call them, higher up in the ocean tube worms. Those alvinella worms are wonderful. They have been observed to grow, I believe, at temperatures all the way up to about 80 degrees Celsius. They can exist in vent fluids about that warm. And although the temperature we're measuring at the moment there is a relatively mild 20 degrees Celsius, they're pretty adaptable to a wide range of temperatures.
We have about 5.5 more minutes for this filter.
Roger.
And one of the neatest adaptations that we have discovered in these tube worms since these vent sites were first discovered in 1977 is that they form these very deep symbiotic partnerships with the microbes that are growing in these environments. And it kind of leads me on to another question that we had on Nautilus Live, which is, how does chemosynthesis work? Which is the description of the type of metabolism these microbes operate. I didn't know if you guys in the back row had a moment to maybe contribute there. If not, just say.
Hey Amy, are you able to give me like an estimated volume filtered?
For the first one, it was 2.77 liters.
Well, for this one right now.
For this one?
Like currently.
I'll say 2.
Okay. It's just requested to give me estimated volume filters throughout the 20 minutes.
Okay.
Where was that request from?
It's in the dive plan.
Okay.
Flow rate's holding steady at 0.7.
IFC team, this is Lounge. Just letting you know that a message has been transmitted via science chat that requires your look-see and some thought, and let us know what you think. Over and out.
Okay, we'll go take a look and we'll respond.
Darlene, do you need my input on that? Samey.
It'd be good to get your input and Eric's input, but we didn't want to bother you right now. Over.
Okay. I wasn't able to access side chat.
Nicole and Sarah can fill you guys in once you're done. Over.
Okay.
All right.
Yeah, so Amy, it was basically what I described to you guys about the change in dive length. Yeah. If you have an opportunity to take a look, it is on this monitor, but I understand if you can't right now.
Okay, thanks. 2 minutes left. Thermocouple's down to 12 degrees. Super is at 7.4.
So maybe just a last quick question from Middle College High School. I know that your biology class has been posting a lot of questions to us. How does chemosynthesis work? Well, as a non-biologist, my quick answer would be, if you think about photosynthesis, a plant or a bacterium can use the energy in sunlight to break apart CO2 and carbon dioxide and water molecules to bring them together then later as sugars. In chemosynthesis,
You want to come down a little bit?
that energy is provided by the chemical potential of some of the minerals contained in this vent fluid. That provides a little jump in energy needed to break apart the basic molecules such as
5 meters at 100.
such as carbon dioxide, water, and
Bridge nav.
oxygen. So really, the question is, where does the source of energy come from? That's the base of the metabolism.
5 meters at bearing 100.
In photosynthesis, it's sunlight. In chemosynthesis, it's the energy contained in some of these minerals in the vent fluid.
Okay.
100.
OEM, I'm going to go. Yeah.
Thermocouple's 15.8. Super's at 8.2. Okay, 20 minutes is up.
Roger.
Pausing. Move to half port. Okay. Data, I got 0.11 liters per minute and 2.27 liters total.
Say that again?
2.27.
2.27 and 0.11 liters per minute. Got it.
Okay.
Maximum temperature for the thermocouple was 21.6.
Okay, great.
Okay.
So next on the agenda, are we going to stow the wand, or are we going to be running some more?
I have one more filter.
One more filter. Roger.
Yeah, and then after that I have 2 bags to fill. I'll flush them 3 times, 30 seconds in and out, and then I'll fill them. But they go really fast. It's only 5 minutes to fill.
Okay, so keep the wand here. And
Yeah, so we'll do this one last filter. So just hang out and look at the pretty
Is it
animals.
S3?
Roger.
Yes, it's S3.
Yep. Yeah. So moving to port S3. Okay.
Thermocouple temperature is at 17.
Beginning filter now. Flow rate's 1.5, 0.7, 0.4, 0.7. Okay. Okay. Okay.
So that was a pretty quick changeover to the next filter. Did you just kind of flip over to the next one over in the rosette?
Yes, that's correct. There are, I don't remember how many ports there are, but I have 9 of them. So I'll have 6 filters and 3 bags, which is the 9 different ports. And then in between each port, there's a half port, and that one is for purging. So if you have any air bubbles, like when we first get in the water, I put it on a half port and then start running the super, and it will push all the air out.
Okay.
Which is important as you go down. You don't want all that air stuck in the system.
Yeah, certainly.
Yeah, and then also when the air comes out, seawater will come in to replace it, and you don't want to get that seawater signal on, like in your DNA sample, for example.
Right.
So the more air you can get out before you even set it down in the water, the better. So we usually use distilled super, not SUPR, but the other super filtered water that doesn't theoretically have bacteria in it.
Right.
So we'll fill up the whole super sampler and all the tubes and everything, and then there will inevitably be some air bubbles in there.
Yep.
So as soon as we get down the water, we try to purge that out. But sometimes when the flow meter slows down, it could be because there's an air bubble stuck somewhere and it can't flow, as fast as it should, or there's enough biomass that's stuck on the filter that it just can't get the water through as fast as normal.
Yeah.
But yeah, I'm not sure why in this case the filter or the super flow is running a little bit slower than it did on our first dive.
Right.
Probably about half as slow. I'm not sure why that is, but once we get to the bags, it's much faster. So you'll see definitely that the wheel is spinning a lot quicker.
Right. So I heard you mention at the end of the 20 minutes of filtering, you collected just over 2 liters of water.
Yeah.
What do you do with that water when it gets to the surface? Because you talked a bit about how you'll study the filters themselves for any trapped microbes. But what about the water you collect?
So we don't actually collect it. It comes out of the exhaust port.
Okay.
So it just comes in and goes through the filter and goes right back out.
Right.
And then that exhaust port is also the intake for the bags. So that's why we switch wands in between the filters and the bags.
Okay.
And then we run the pump in reverse and it fills up the bags. Because
Okay.
there's not an inlet and an outlet for the bags. There's only an inlet. So
Right.
that's why we have to switch to the outlet and just go suck it right up.
And then what will you do with the water that you collect in the bags?
So we have lots of experiments planned. We are doing activity measurements. We want to know which organisms are fixing carbon. We have some very intricate experiments to test that. And then Sarah's doing some grazing experiments to see what like very small eukaryotes will graze on the bacteria that are present in the fluid.
Okay. Oh, I remember that one. Is that the one where you
Yeah.
dye them with a fluorescent dye and then you
That's
see how that
correct.
dye is taken up in the eukaryotes?
That's correct.
Right.
Yeah.
I
So
paid
she's
attention.
doing that. Yeah. And then I'll also start some cultures. I have a variety of different culture media to try to enrich for certain microbes that either reduce sulfate or use hydrogen and CO2. I'm really interested in acetogens because they can fix carbon and use hydrogen as an energy source.
Uh-huh.
And, we found hydrogen on Enceladus
Yep.
and potential hydrothermal vent location. So that's what I'm most interested in trying to enrich from this area.
Right.
But
And those acetogens are quite... so what we mean by acetogen is they metabolize, is it acetic acid or is it acetone?
they make acetate.
Oh, they
Yeah.
make acetate. Okay, thank you.
Yeah.
And but essentially in their metabolism, they don't use the molecular oxygen that's dissolved in seawater,
No,
do
they do not like oxygen at all.
Right.
So some of the enzymes that they have to use in these pathways are, the enzymes are inactivated in the presence of oxygen because it's such a strong oxidant that it kind of screws with their metabolism.
Yeah, it's a highly reactive molecule.
Yeah. So we believe that that type of metabolism is really ancient and was here on Earth prior to the Great Oxidation Event. So they're kind of primitive, very simple organisms, which is also another reason why I'm really interested in their metabolisms.
Right.
And they can form the base of the food chain because they make acetate, which is a really important key biomolecule for almost all biosynthetic pathways.
Okay.
Yeah. So it's also very similar to methanogenesis. It's just a few different enzymes than methanogenesis.
Okay.
But it seems like this acetogens might have come a little bit earlier because it is a little bit simpler.
Right.
And based on some other predictions that people have made based on organisms or life that could live or begin in like pockets of rocks and hydrothermal areas and things like that, it seems more likely that acetogens would have been the first types of life that arose on Earth. But, you know, it's all speculation. We don't know that for sure, but I definitely want to try to get some of these organisms in culture and see if I can culture acetogens from vents. That hasn't been done before.
Oh, wonderful.
Yeah.
So are these acetogens, you know, part of the family of microbes that we call archaea, that are separate to bacteria, or are they a bacterium?
These are bacteria.
Okay, interesting.
Data,
hey Alex.
sorry, Data, do you have the time that I started that? Because my timer stopped.
Yeah, no problem. 1829 UTC.
Okay.
Alex,
So it's been
let's
6
do
minutes?
another 5
Okay.
meters at 120.
120, copy.
Thank you.
Bridge nav.
Thermocouples at 18.1.
Yeah, 5 meters at bearing 120.
Super temps at 10.7.
What I thought was really neat, Amy, is when I toured your lab and Sarah's lab on board the ship, is it really brings home that there's no one experiment or test that you do to study these life forms, these microbial life forms. You need multiple different tests to kind of give you each a different perspective on what their metabolism is or their genetic properties. It's a very complementary form of research.
Right, right. So we're generally interested in who is there, what are they doing, and how fast are they doing it. And so the DNA analysis will kind of give us an idea of who is there and what types of genes or pathways that they have. And then the RNA analysis will tell us who's active and who's actively doing what and using what enzymes. And then we want to know who's fixing the carbon and how fast they can do it as well. So we have to do some experiments with that as well.
That was a neat one. I remember you describing that to me. You basically feed them a source of carbon that's been labeled with naturally radioactive carbon-13, and then you see how they process that. Is that right?
Yes, that's correct.
Yeah.
Thermocouples down to 9.3, and supers at 8.2.
So I see just from the science log that this vent is called Ventilate.
Yeah.
Are you kind of saving Grande for anything we might discover over the next?
We were going to do soy Ventilate too. If we had a series of vents, we had a whole bunch of them. No whip. Yeah.
So you guys are primed for discovery.
Yeah.
Amy, can you give me like an estimated volume filter?
Yeah. Let's see.
Is the flow rate like pretty consistent?
I think so. It's around 0.7.
Okay.
Let's see.
It's kind of too bad we can't tell what these
So then probably like 7.
tube worms are thinking or anything, you know? They're a mystery. They could use a ventriloquist.
It's about 0.12 liters per minute.
Yeah, they might. Well, if they could
yeah,
think,
okay.
they'd probably be thinking, who turned on the lights?
So like 12 liters, I guess, because we're like 10 minutes in. Right?
I can't do math right now. I'm so tired.
Wait, you said so it's 0.12 liters per minute?
Yes, I think so.
Okay.
So it's about the same as the previous one.
Okay.
Kind of looks like the whole arm there has been reclaimed by those little worms around it. They really, everything's kind of come back. I've seen less and less of it as it sits there.
Well, I guess later today we're going to revisit the microcolonizers, and
Yes.
it was really striking because you see them covered in limpets, you know, and so those have all crawled on there over the intervening days.
Yeah.
Thermocouple's back up to 13. I'm sorry, 17. Super's 6.4.
So just a quick update for those of you watching on Nautilus Live. What you're seeing at the moment in the main camera of Hercules that's zoomed in quite far is a section of diffuse flow hydrothermal vent that is part of the wider Seacliff hydrothermal vent field. All those stringy bush-like things you can see, those are the worms of the tubes of tube worms. What we're doing at the moment is currently filtering some of that vent fluid that's coming out in that sort of shimmering haze you can see, hoping to later study any microbes that it contains. If you zoom out from where we are to the much bigger picture, the Seacliff hydrothermal vent field is part of Gorda Ridge, which is the tectonic plate boundary between the Gorda and the Pacific tectonic plates. We're currently operating at a depth of 2.7 kilometers under the ocean, and the Nautilus itself is exploring what we call the Eastern Pacific Ocean, which is, and we're actually located about 180 kilometers off the west coast of North America, just where California meets Oregon.
There are about 7 minutes left.
Roger.
Your psychic powers are amazing, Amy, not just telekinesis, but I was just about to ask that question.
Look like very small trumpets.
Yeah.
We still can't hear them. Maybe they are.
Get cacophony of that.
I imagine if we put a hydrophone here, we'd kind of just get this, I guess, audible equivalent of the gentle shimmering that we're seeing in the diffuse flow.
Only if we could remove Herc's noise.
Yes.
Noisy beast.
No, that's a good point. I sometimes wonder if we did that same experiment at one of the sites of focus flow with the chimneys, whether it would sound like a jet engine.
Yeah, it would be really neat. We've done, so NOAA and the sanctuaries are really interested in soundscapes of the ocean.
Oh,
to
yeah.
determine mostly if there's impacts of human noise, which there are in a lot of coastal environments, you know, boat traffic and all of that. We left a hydrophone at a seep site, and we've done it 2 years now. I haven't heard the results from the second year, but the first year even, I think we were about 1 kilometer away and we could still hear Herc. The scientists were able to remove that signature and hear the bubbles coming out of the seep.
That's amazing.
Which is pretty cool.
Yeah.
Because they're able to then maybe over a longer time period monitor their activity just by listening.
Oh. Okay.
That's an interesting comment, Nicole, because I know that the other way that these methane seeps are studied is with basically seafloor sonar devices.
Right.
Did the hydrophones give you a much greater range so you kind of can monitor a larger area?
Oh, sorry. I'm not exactly sure. I think it was actually giving them sort of a point source. So what you could do is one of these seeps, seeps individually are pretty intermittent.
Yeah.
So you could set it down next to one that you want to know the rate and how intermittent it is, and something about, like its period of activity versus inactivity.
Right.
And then leave it for maybe, depending on battery and storage, weeks to months. Come back, pick it up, and then have an idea of just how intermittent seeps in a certain area are, or a particular seep is.
Right.
Because the sonar gives you, 1 point in time, and then sometimes we'll map the same seep multiple times just because it's a good test for our sonar. So for example, shakedown, our system tests are always off Southern California.
Right.
So there's one seep we like to go just do a couple of passes over to make sure we're still detecting, able to detect seeps in the water column with the ship's sonar.
Uh-huh.
And so, having a hydrophone that you could set down for a long period of time would give you a nice time series.
Cool.
We haven't named that seep, no. I know. There's so many seeps in the ocean. Yeah. No, it is one exact one.
Is that the new there are so many fish in the sea?
I know, it's a terrible thing to say.
So many seeps in the ocean.
This is a very special seep. Actually, next year we're hoping to dive on it as well. Yeah, we have to look at it first.
Can we talk about what exactly is a tube worm? Because I could see them, but are they really worms? What is going on in there? Anybody?
Well, the tubes are made of chitin, so they grow. The tubes, and the worm is the frilly part that you see inside, and really usually it only is, as far as I understand with most of these, the worm tube will grow so that it can stay in the flow and keep feeding, but the tube worm itself isn't the entire length of the tube. And obviously it retracts into the tube when it gets disturbed by something to stay safe. So yeah, you're seeing it kind of sticking its gills sort of out, right? Like it's and they usually host a whole bunch of symbionts that are actually doing the chemosynthetic or chemosynthesis work for them.
Cool.
But I'm a geologist, so anybody feel free to jump in. And
good answer.
2 more minutes. Did you start it right at 20?
But yeah, to answer your question, they are a real worm. It's just in the adult phase, they don't crawl around like you think of an earthworm.
Yeah.
They, at the base, they essentially, they grab onto the rock, and they're there for essentially, I believe, all of their adult life. They're not moving.
Yeah.
And so it's one of the features of understanding hydrothermal vents is that we think they're quite dynamic environments. And so we see when we revisit sites where the vent activity has shifted or changed, and it will be surrounded by fields of basically just the tubes left over as once you take away the food source for, as Nicole mentioned, the chemosynthetic bacteria, the worms that host them will just die as the food source is taken away. And the way they actually travel around the ocean is, we believe, at the larval stage.
Yeah.
When they reproduce, they produce larvae, and they float freely.
Yeah.
And then the ones that take root in the vicinity of a hydrothermal vent, those are the ones that will survive.
Yeah. Kind of like little seeds.
Yeah,
no,
exactly like that.
Just how a lot of things in the ocean and water are dispersed.
Yeah.
You just use the water, which makes total sense.
I think Herc's arm has a squat lobster on it. On the tube. It is being totally colonized. Just not going to be able to pull it out afterwards.
Are we getting close to the end?
Yes. I can go ahead and stop.
Okay.
Temperature on the thermocouple is 18.6. The super is 8.7.
Okay, stopped.
Okay, I'm going to stop recording the thermocouple.
I'm going to advance to half port.
What was the max temp?
22.9. Okay.
And then when you get a chance, the final volume and flow rate.
Yeah.
Okay.
1 second.
So just a quick time check for those of you following. It's just coming up to noon Pacific time. In just a few minutes, you'll hear a little bit of activity in the control van as the...
Data?
Yeah. Yep.
Flow rate is 0.14 liters per minute.
0.14, got it?
Yes. Volume was 2.8.
Okay, 2.8 liters.
Yeah. Are you ready to advance to back?
Yes.
Okay. We're going to advance to port 7. Did you need something? I'm just checking.
So yeah, over
We're
the
filling
next...
2 bags.
Yes, we're going to
Flushing
fill 2
them
bags
first.
here.
No, two and then 1 later, I think.
So over the next 10 minutes or so, some of the communications will get a little bit intermittent as each watch member hands over to their counterpart. So bear with us as we go through that process.
Okay, so I'm at port 7. I'm going to fill for 30 seconds and then flush for 30 seconds and do that 3 times. And then I'll fill for 5.
And Amy, this is confirmed. We're still using this wand.
Oh, no, sorry. We need to switch to the other wand. Yeah.
Roger. So am I clear to stow this one?
Yes.
Roger.
Thank you.
Video, you want to come wide, please?
Yep. Those worms are going to have to let go of it.
This is going to be a bit of a challenge.
What?
Yeah.
To do this while we're sitting here.
Yeah. Woohoo, yeah, well.
Yeah. So, can we just talk about the few steps ahead of where we are right now? So we want to stow this wand, pull the other one out, and put that into the same spot that this one's in?
Yes.
Okay.
Roger.
So we have to move to do that. We can't get up. We can't, as you can see, the porch is overrun with worms. So yeah, we're gonna have to come back to this spot like we did before.
It works.
Okay.
I have an arm print to fit right into.
Yeah.
Roger.
Do you want to? What do you want to do? Do you want me to fly back and you do the?
Sure.
Yeah. Yeah, go ahead and pull that out a little bit. Just be careful of your elbow. I'm sure you're going to get right up that bumper bar pretty quick.
Roger.
Okay. I'll come off.
I'm just going to tilt down.
Okay.
Would you mind giving me the porch?
Yeah. I just put it on the magnet.
Roger.
Yeah, exactly.
You want it face in, correct?
Yeah, sure.
So right now we're just stowing this particular wand that's used to feed fluid into the super sampler, and we're just going to replace it with a second intake tube that's necessary to actually retrieve some water samples as opposed to simply filtering it.
You can do it any which way that's easiest, doesn't really matter.
Roger.
If you want to face it outward, it's fine too.
Hello.
Hi.
So welcome to the noon to 4 watch. We're going to be doing a little bit of a change over the next few minutes.
Sorry. I'm just going to store it outwards like this. Oh, under the porch. Everything's happy. There it goes. Oh, we have a...
Chris, are you at the ISC?
There's a bunch of us here.
Hi, Chris. I was just wondering if you've been back to that taco shop. I don't know what it was called. That's all I can think about every time I sit down for dinner. I'm like, I wonder if Chris is eating tacos.
I've been resisting the taco shop.
You did go?
Negative.
Oh, negative.
Been sitting in this chair all weekend waiting for you to get back to the seafloor, so I haven't had time to go to the taco shop.
Okay.
Amar handcuffed me to the chair and said, you will stay at the ISC.
So they're about to do a shift change. There's like 20 people in here.
Okay, ISC standby. We're just completing our watch handover. I think they're just going to relocate the ROV back down into site, and then they will complete the handover of the pilots as well. Okay, vehicle is now in position. We're just waiting for our Herc pilots to do their handover. Standby.
Hello, world.
Hi there, Trevor.
Hi, Lee.
Hello.
Hi, Lee.
Yeah, I'm the world. Just me. Hi, everyone.
4 is on.
Okay, so is everybody on headsets? Everyone completed their handover successfully?
Yep.
Believe so.
Excellent. Great news. Okay, Trevor, I'm going to hand over to Amy because I believe we're going to go for a super bag. Is that correct, Amy? Yes, that's correct. Okay. I think, are you on SPL, Amy? Are you on?
You're my
Nope.
worms.
No, you're not.
Yeah.
Oh.
Am
And
I
again?
now?
Yes,
There
you
you
are. We can hear you loud and clear.
Sorry.
Brilliant. No worries, Amy. Thank you. So yeah, Trevor, I'm going to hand over to Amy.
Yeah.
Sounds great.
So we're switching to the super exhaust, which looks like you have.
Got it.
And the tricky part is finding the spot that we were in before.
Yeah, I got a bit of a handover on that, so I can
Oh,
get it kind of
good.
close, but I'll defer to you for the final thing.
Okay.
What's going on there?
I don't know.
Nice.
Got some worms in the way.
So, let's see here. How am I going to do this? Yikes.
Oh, look at that fish.
That is too high. Okay, so I think where I'm going is right about here-ish, Is that true? Does that seem reasonable?
I think you need to come forward.
And a little bit lower.
Yeah.
Forward isn't closer to me or away from me?
Closer to you.
Closer to you.
Okay. And down
and
here?
Down, yeah.
I might try to avoid this little side bush bit. Like this zone here?
I think that's too deep. Maybe.
Yeah, I didn't get an exact spot, so I'll definitely.
Do you see
That's getting a little closer.
The left side of the field of limpets in the center there, the red part?
Okay.
I think it was, yeah, very similar to right there because it was along the edge there.
Okay. Let me reposition a little bit here. Like here? We have a pointing stick up here. Feel free to come up and point it out to me if you know the exact zone. Sure, that's that pointing stick. Pointing stick of science.
Yep.
It's the most scientific way.
Sure. Down there. Okay, got it.
All right.
So like, let's try there. Is that a happy spot there? We can zoom in a little bit, please.
Yeah, that'd be great.
Happily.
Good there. I can wiggle it around a little bit just so you know where I'm at. Understood. Yeah. I think you're off SPL again.
That looks good.
Check out that little flaky bit there. Ah, I got a flaky bit stuck on the end there. Get off of there. Oh no. Okay, well, oh, it's gone. Okay. Medium.
Oh, no,
medium.
there. It's just longer.
Just collected more sticky bits.
Okay, is that a good spot to lock it down?
That's a pretty good spot. Do you think so, Sarah?
Yeah,
okay.
It looks good.
I'm going to move to the port 7.
Okay, we are stable there. You got camera on bag.
Yeah,
can we get the
put
camera?
The flow meter on? Sorry.
Yep.
You got flow meter, sure.
Do we want the bag on the quad? Is that what we're doing?
ISC, do you want the flow meter or the bags?
Could we put the bags on sat feed 2?
Okay.
Sure. Yeah. So we can have the flow
That'd be
meter
great.
on 3 and yeah, the bags on sat feed 2 if possible.
Okay.
All right, bags
There we
on
go.
feed 2.
Thank you, video. That's great.
You're welcome.
Do you want a little more zoom in on the flow meter?
More zoomed, the less focus.
Okay.
But it might
Sorry,
work.
they've got
actually, that's perfect. Never
Yeah.
mind.
The heat coming through.
I'm just moving to port 7
That looks
now.
good. Thanks. Filling bag 7.
So I'm at port 7. So we're gonna do 30 seconds in and 30 seconds out to flush it. We'll do that 3 times and then we'll fill for 5 minutes. And then we're gonna do 1 more of those bags. Let me get my timer set. Okay, start now. 30 seconds fill.
Flow meter looks good, so it was just the filters from previous.
Oh, I did 30 minutes. Thanks for taking care of me, Sarah.
I gotcha.